in a college full of aspiring scientists, insane professors, and wacky lab managers.
KD squared, that's us, and our classmates try to do the impossible. Create a guide that will help you survive cell biology.
Luckily, both Kennedy and I have taken Biology 4411 and understand the importance of cellular respiration. Now you might be thinking, gee, that's great for you, and it is, but don't worry, we're here to share our vast knowledge and appreciation of this vital series of metabolic processes.
The first step is understanding what cellular respiration is. Cellular respiration is the process by which biological fuels, mainly glucose, are oxidized in the presence of an inorganic electron acceptor, such as oxygen, to drive the bulk production of ATP.
Cellular respiration involves three crucial steps, the first of which being glycolysis, the second, the Krebs cycle, and the third, the electron transport chain. For our purposes, we'll be focusing on the Krebs cycle, also referred to as the citric acid cycle or the TCA cycle.
The Krebs cycle is a circular series of reactions that take place in the mitochondrial matrix. These reactions release the energy stored in nutrients through the oxidation of the acetyl-CoA into two molecules of carbon dioxide.
The citric acid cycle in eukaryotes takes place in the mitochondria, while in prokaryotes it takes place in the cytoplasm. This cycle only occurs under aerobic conditions, as energy-rich molecules like NAD+ and FAD can only be retrieved from their reduced form once they transfer electrons to molecular oxygen. When oxygen is present, acetyl-CoA is produced from the pyruvate molecules created in glycolysis.
The first reaction of the Krebs cycle is the condensation of acetyl-CoA to citrate by the enzyme citrate synthetase. The methyl group of acetyl-CoA is converted to methylene in citrate.
The second step is the conversion of citrate to isocitrate by the enzyme aconitase. This is a dehydration-rehydration reaction in which water is a product when converting citrate to cis-aconitate, and water is a reactant in converting cis-aconitate into isocitrate.
Isocitrate is then converted into alpha-ketoglutarate by isocitrate dehydrogenase. This process occurs through oxidative decarboxylation, in which the hydroxyl group is oxidized to a carbonyl, which facilitates decarboxylation by stabilizing the carbanion formed on the adjacent carbon. This reaction is also one of the two in the Krebs cycle that produces a molecule of carbon dioxide. This is the first of the three steps in the Krebs cycle that reduces a molecule of NAD+ to NADH.
Alpha-ketoglutarate then becomes succinyl-CoA by the enzyme alpha-ketoglutarate dehydrogenase. This is another oxidative decarboxylation reaction that produces the second and last molecule of carbon dioxide from the Krebs cycle. This reaction, like the previous, also reduces a molecule of NAD+ to NADH.
Succinyl-CoA is converted into succinate by the enzyme succinyl-CoA synthetase. This is a substrate-level phosphorylation reaction in which the energy is given off in the form of one GTP molecule.
Succinate becomes fumarate with the help of an enzyme called succinate dehydrogenase. This is a dehydrogenation reaction in which the introduction of a double bond initiates methylene oxidation. This reaction is the only time in which a molecule of FAD is reduced to a molecule of FADH2 in the Krebs cycle.
Fumarate is converted into malate by fumarase and the addition of water. This hydration reaction introduces a hydroxyl group for the next oxidation step.
Finally, malate becomes converted into oxaloacetate by the enzyme malate dehydrogenase. And you guessed it, this is a dehydrogenation reaction. Oxidation of the hydroxyl group generates a carbonyl group that is ready to facilitate the condensation of the next step, which just so happens to be the first step of the Krebs cycle. In this reaction, we get our final molecule of NADH2, and the cycle is ready to repeat itself.
Each turn of the Krebs cycle produces one GTP molecule, 3 NADH molecules, 1 FADH2 molecules, and 2 carbon dioxide molecules.
The GTP molecule can then be used to produce ATP for further energy use. The NADH and FADH2 molecules will be used later in the electron transport chain. Carbon dioxide ultimately needs to be removed from the body by transportation out of the lungs.